Herbicide tolerance is the inherent ability of a species to survive and reproduce after herbicide treatment at a normal use rate. There is no selection involved because the species is naturally tolerant. For example, glyphosate and glyphosate. The influence of herbicide in agriculture has raised, and the call for herbicide-resistant crop production is also increased. For that, dating has opened new avenues to accelerate herbicide-resistant crop development. Genome editing offers several advantages in herbicide-resistant development in various ways. For example, genome editing enables the direct insertion of an exogenous herbicide-resistant gene in a plant to confer herbicide resistance. More importantly, genome editing, especially programmable base editing, presents heritable targeted modification leading to a transgene-free herbicide-resistant crop that are genetically non-distinguishable from that of the plants created by traditional mutagenesis. Moreover, base editing offers multiple amino acid substitution of targeted genes that confer resistance to multiple herbicides. Notably, unlike genetically modified plants, the non-transgenic plants produced via CRISPR-Cas system are free from any additional regulatory approval in several countries. Therefore, genome editing can expedite the commercialization of herbicide-resistant crops. Compared to mutagenesis and genetically modified approach, genome editing can save the time and cost of herbicide-resistant development, as the genome-edited herbicide-resistant crops generated throughout the introduction of foreign DNA do not require a risk assessment. This allows developers to launch new crops into the market years early and for millions of dollars less than genetically modified crops.
Next, I will present the disadvantages of herbicide tolerance. First, it will increase herbicide use. There seem to be two major explanations why herbicide use in herbicide-tolerant crop may increase. One reason is that a high level of crop tolerance may enable the farmer to increase doses to achieve and improve weed control without harming the crop. The other reason is problem with tolerant weeds and volunteers, which requires farmer to increase dose or mix herbicide with different modes of action to maintain an acceptable level of weed control, which means it continue our dependence on herbicide and it really didn't change anything. It is just changed which herbicide we were using. Next, the application of this technology have certain limitations, such as edited herbicide-tolerant crops are considered as genetically modified in some countries that are subjected to strict regulatory procedures. In the case of genetically modified, the major factor restricting the production of herbicide-tolerant plants include the higher cost to obtain regulatory approval along with international trade issues. Therefore, due to this limitation, the advantage of robust technology to rapidly develop and commercialize genome-edited herbicide-tolerant crops can be restricted. Moreover, the society's concern about the genome editing is in part due to a lack of knowledge of its application and principles. The primary feature here is the difference between genome-edited, genetically modified, and transgenic plant, and the plants produced through the genome editing may or may not be transgenic. Therefore, currently, the public education about genome editing principles and the knowledge of its difference from older breeding techniques is necessary for correcting and preventing the spread of suspicion and misconception. Lastly, gene flow from crops to other crops or related species in another route to the development of resistant weed population in the field. Once the resistant gene is present in the crop volunteers or related weed species, then it is expected that the same weed control practices which cause herbicide resistance to occur in naturally tolerant wheat biotypes will lead to a rapid build-up of herbicide-tolerant wheats and volunteers.
The type of genome-edited herbicide resistance crop in agriculture is herbicide resistance crop consistently represents the largest planting area of biotypes crop from 1996 to 2018. Herbicide resistance crop occupied 87.5 million hectares in 2018. Glyphosate and glufosinate tolerance cultivar are the most frequent. For instance, the countries that have approved a major herbicide resistance crop for food, feed, or cultivation is Australia, plants soybean, which are the herbicide-tolerant crops. The usage of herbicide-resistant crop benefits the environment, according to the recent study undertaken by the Council for Agricultural Science and Technology. Since the introduction of herbicide-resistant soybean in the United States, novelty soybean has expanded by 35%. Furthermore, the regions entirely dependent on glyphosate-resistant crop led to gradual rise of glyphosate-resistant weeds. To address these constraints, genome editing has been used to generate a number of non-transgenic crops, just a handful of which have recently been marketed. For instance, soybean applied CRISPR Cas9-mediated genome editing technology. The fact that genome editing has been used to generate a number of crops, agricultural trials of the crops are limited. Species that may be prone to glyphosate resistance based on resistance to other herbicide modes of action, including waterhemp, common, and giant red wheat. Moreover, the use of genome-edited herbicide-resistant crop with multiple herbicide resistance can be a key strategy for combating resistant wheat, increasing crop yields as well as soil moisture and preservation. As the development of new herbicide mode of action would give growers more options for weed control, multiple herbicide-resistant crops were created by stacking tolerance rates for various herbicide chemicals. For instance, soybean is glyphosate and dicamba tolerance. This technology is expected to bring to the weed management toolbox a new tool for controlling resistance and difficult to control weeds. However, uncontrolled uses of relying on this technology could lead to more complicated weed problems, including various herbicide-resistant weeds.
The process of creating herbicide-tolerant organisms starts with the PCR of the gene of interest. A study from African Journal of Biotechnology shows the method of amplifying herbicide-tolerant gene in soybean through PCR. From this journal, the herbicide-tolerant gene for soybean found are called EPSPS gene. It starts with the addition of primer with PCR component before PCR cycling begins. Primer functions as a marker for the DNA polymerase to copy the DNA region of interest. In this experiment, this is the forward and reverse primer used. The primer will stick to the end of the gene to transcribe the same copy of gene multiple times. The condition used for PCR is as shown here. This process amplifies the number of EPSPS gene that is required to be added into a host before it is able to create the herbicide-tolerant organism. Afterward, the gene is added into a plasmid DNA to form recombinant plasmid DNA. That recombinant plasmid DNA is then inserted into the competent host before it can be transferred into the soybean. As a conclusion, the herbicide-tolerant organism is a part of genetically modified organism or GMO. Although this creates the opportunity to alter the gene of the organism in the manner that is beneficial to the consumer, GMO also cause some trust issue in people due to the unnatural side of it.